Darren.
Right. Welcome, everyone. I like it, seeing this place full. We might have to find a larger meeting room next time. It was great seeing you all. I have the pleasure today of introducing today's speaker, Dr. Zachary Fox. He's a research scientist in machine learning for and artificial intelligence in health at Oak Ridge National Labs. He got his undergraduate degree, Bachelor's of Science in Biomedical Engineering from University of Delaware. Did his PhD with Brian Munsky at Colorado State in Fort Collins. Went on to do his first postdoc at the Institut Pasteur in France, but it was cut short during COVID, unfortunately. He moved on to Oak Ridge National Labs. I'm sorry, he moved on to Los Alamos National Labs for his second postdoc, and then wound up at Oak Ridge National Labs, just down the road from us here in East Tennessee. And looking forward to hearing his talk about machine learning and how he's applied some of that to well, first he's going to talk about his optogenetics studies, that the work he did in the Institut Pasteur, and then he'll talk about more of his more recent stuff in machine learning that he's been doing at Oak Ridge National Lab. So, thank you.
Cool. Well, thanks so much for having me. It's really great to be back at Vanderbilt and hanging out and meeting lots of fun people. Let me fix this now. I've had an awesome visit so far. Thanks everyone for coming to listen to my talk. And I've labeled it Adventures in Machine Learning for Biomedical Science, which is like obviously a super broad topic, but I'm gonna be doing probably more systems biology and experimental stuff during the first part of my talk, and then I'll move into some more machine learning applications later on. The images are generated by the Stable Diffusion model with the prompt machine learning for biomedical science. And so I thought those were some fun and cool images. And yeah, let's get started. So Darren kind of gave you all the short version of my path to Oak Ridge National Lab. I just started there in October of 2022. So that is in kind of the way, don't you think? How do there's a way to hide this whole thing.
Floating more. No, go to more again. Go down. Yeah.
This one?
Yeah.
Thanks.
Yeah.
You would think. Okay. So as I'm sure you all are aware, we study biological systems at a bunch of different scales and a bunch of different ways. And what I'm going to be talking about today is mostly the first part will be like single cell kind of studies in yeast. And then at the end, I'm going to talk a little bit kind of at the molecular scale in talking about protein-protein interactions. And but I think like the sort of methods and ideas have been applied across a variety of scales, and hopefully translate across scales. So when we think about doing experiments, I kind of want to set up the discussion and the presentation by giving kind of a broad view of how I view quantitative biological research, and kind of where the things that I'm interested in fit. So obviously we have experiments, we need data. That I have a microscope, but it doesn't need to be microscopy data. You measure what's going on in the cells, and you analyze the data using statistics and different types of approaches. And then a thing that we like to do as quantitative scientists is kind of encode what we think about is going on in the system in some set of equations, some kind of logical equations. They could be ODEs, they could be different types of modeling approaches that all are valid, depending on the problem. And then we want to kind of calibrate those models or that logical way that we're thinking about the problem from our data, and then use that to predict what we think will happen under different experimental conditions or something like that. And then if we have a good model, we can use it to help design future experiments. And that experiment design could be, I want to make data that's going to give me a model which can lead to better predictions, and you have this kind of looping idea. It might be you want a model which you can just predict well with, so you can control your system to move away from cancer phenotypes and into healthy phenotypes, things like that. But I think like having this model and actually writing down the set of equations that describes like what you think about this is going on in a system is a super important step. And I also wanted to take a second and just say, if anybody has questions or something's not clear throughout, just interrupt me during rather than waiting till the end. So yeah, this is the outline. I'm going to be talking about using models to predict and control the gene expression in individual cells under a microscope. And then I'm going to be talking about using machine learning to predict protein-protein binding affinities and some of the challenges that we face on that problem. So this first part falls under what people are calling single cell cybergenetics. And this is kind of a glamorous way of saying we want to interface cells with computers. So just like the way that we can think of like a you know a robot and a human cyborg, we kind of want to have like cell cyborgs. Like you have a computer that can control the biology, and the feedback can then affect what is happening in the computer, and so on and so forth. And so we need a set of tools to be able to do that. And people have been kind of thinking about these sorts of hybrid biodigital systems in a number of studies for a handful of years. People have done things like build oscillators using optogenetics. They can study feedback mechanisms by deleting, creating mutant cells which don't have feedbacks, and then reconstituting the feedbacks with computational control algorithms. Or they can try to control dynamics of transcription in single cells, which is going to be more in line with what I'm going to talk about today. So what are the ingredients for this kind of system? So the way that we built our system was using this EL222 optogenetic protein. It comes from bacteria, but it's been put in yeast and is pretty robust in that system. So basically, the idea is you shine blue light on the cells, and it causes this transcription factor to dimerize. When that transcription factor dimerizes, it can go to a promoter and drive the gene expression of something. And when you turn the blue light off, the molecules unbind, and the transcription will no longer be activated. And so we did sort of a pulse width modulation experiment where we applied different amounts of light to different cells, kind of varying the width of the pulses, which were sent every 3 minutes. And we saw that we can get more or less fluorescent protein production in those cells kind of based on the pulse width. So we can actually grade the amount of gene expression in those cells. We need a way to shine light onto individual cells. I'll talk more about this. So we have a digital micromirror device, which is basically just like a projector, which has a bunch of little mirrors, and they tilt. And so if you're shining a light at them, depending on which direction they're tilting, they're either pointing at your sample or not. So you kind of have this pixel-wise resolution of a growing population of cells. You need a microscope, microfluidics, got to keep your cells happy and alive. And then you need software and algorithms. You need ways to basically take your bioimages and your microscopy images, process them, and feed them back into your control algorithm or whatever it is that you're trying to do to the cells. And a lot of the work that I'm talking about was put into kind of this component. We sort of were able to use pre-existing synthetic biological systems. So this is just a simple movie of different of a yeast microcolonies growing under the microscope. And we're just shining light on them, and you can see the red fluorescent protein being expressed. And one of the key, you can see that you have pretty different expression in different cells. So this could be due for any number of factors, but we had tested pretty well that the illumination was equal across the population. So this is due to different biological variability, whether it's at this dimerization of the molecule or at the promoter, so on and so forth. Okay, so I've showed that we can build a system where we can kind of shine light on individual cells and turn on gene expression. Okay, and now I want to think about this as a sort of classical control engineering problem, where you have a set point, which is our number of molecules over time in each individual cell. We have a way to actuate, so we have a digital micromirror. We have the plant, which would be our yeast cells, a way of measuring, so that's our microscope, and then the output would be the sort of number of molecules in a cell over time. The questions we wanted, some of the questions we wanted to answer was like, how do you control? What does the controller look like? How do you determine what light to send to cells to get them to have this profile? And how do you build the feedback? How do you interpret the data to put back into your controller? I kind of described this already, but here it is again. You have, just imagine these cells all have this EL222 gene, which is constantly being expressed. So it expresses the monomer, and we can model that, which I'll talk about later. And then you shine blue light and they dimerize, and when they're dimerized, they activate the fluorescent protein. And then we have this digital micromirror device. And so this is an image of what they look like. So you can see this mirror is tilted. We can control each mirror independently. And basically this allows us to shine light on different parts of the field of view with subcellular resolution. We also need to be able to segment and track cells in real time, because if we want to do a control experiment, we need to take a picture of our cells. We need to know which cell is which, because we want to basically control each cell individually, so they need to have an individual identity. And then we need to do that kind of quickly. Okay, we are segmenting and tracking every 3 minutes. So we built a tool. This was kind of, I don't know how many folks are doing cell segmentation, but these days there's really great algorithms that are kind of off the shelf that work. This was slightly before that was available. And also those things tend to be a little slower than what we needed. So we trained our own UNet. So this is a neural network architecture, which is really good for segmentation and has been shown to be like pretty much state of the art, even with all the fancy new models that exist now. This kind of architecture still does great. These aren't particularly challenging segmentations, okay? They're just circles under the microscope. But we built a training data set, and small over there on the right, you can see that we can segment a field of view with just a few cells or with a densely packed layer, monolayer of cells, efficiently and quickly, and they're well separated. I forgot what else I was going to say, but yeah, so you can do segmentation, and these things are again was trained on our data, and then we can track the cells as well algorithmically. And this is just using a classic tracking algorithm, because once you've developed the segmentation, the tracking is really not so hard. You have a few challenges in our system, which was you have this new cells being born and you need to detect them. So you have kind of this mask, this cell was detected, but it was maybe starting to be born a frame earlier. So it can be challenging to miss. It's possible to miss some cells. Like in this picture, you can see there's probably a baby there that we missed. This highlights that the tracking is really not necessarily the hard part. it's more the segmentation and getting that really good. If that's really good and works when you just have newborn cells, et cetera, then the tracking is relatively easy to do because these things aren't moving around too much.
So when you do so you said you image every 3 minutes,
Mhm.
does it mean bright field and presence?
Yeah.
And can you actually do bright field imaging faster?
So that's a good question. So we were, we actually did the bright field every 3 minutes because that's what we needed to track the cells accurately. And we did the fluorescence every 6 minutes. And was because, that was kind of we didn't need to go faster for the bright field for tracking. And we didn't want to have like photo bleaching and that toxicity and that kind of stuff with too many images. So
Okay.
Yeah.
Because bright field, I mean, you don't use a lot of light.
yeah.
Yeah.
Yeah, so I think we
So-
definitely could image faster on the bright field.
It's not necessary.
But yeah, there was not really much point. The other thing is like, we wanted to segment in real time, right? So, but actually like even that's what I was gonna say, this super dense field of view, segmenting hundreds of cells was less than a second to do. So it really was not a bottleneck. We could have in principle measured quite quickly. And we made sure everything was good. So we checked our tracking and segmentation algorithms by hand, which was super fun. I don't know if any of you guys have been down that road. It still beats, I guess, like doing all of them by hand all the time. But and so we basically saw kind of different types of tracking errors, like the cells flowing away was a big issue in the microfluidic channel. And which was kind of like it's a tracking error, but it's not. It's not something we could do anything about really, besides make our microfluidics better. But there were real tracking errors. So here you had like the buds tend to like kind of rotate along the outside of the cell, especially when they're isolated like this. And for whatever reason, it moved far enough between frames that we lost the track on that particular cell.
So in the microfluidic, can the buds also go upwards, or is it that?
No, it's like a monolayer. Yeah. It's actually the chamber was like kind of flat and then like sloped. And so where the cells were was, whatever height they could go this way until they stopped. But yeah, we saw we had pretty accurate track. Like this was good enough to do what we wanted to do. We can track the cells well. And then we spent a lot of time developing software to like basically do all of these things in general. And I say we, but it was more or less Stephen, my colleague at Pasteur, who was like the engineer who did a lot of the software development for this image analysis. And it was really a general microscopy pipeline for if you have some imaging event, whatever you want, you can define these kind of events and they have triggers and effects. So you could analyze your image, which would say, for example, like check the number of cells in your frame. And the effect could also be something else you can control Pythonically. So this was all a extensible Python library. You can write your own modules easily. And it also kind of interfaces with like your hardware. So if you had a microfluidic pump that you wanted to attach, you could make an effect which changes your microfluidics, etc. We also hooked it up to Discord and like made it so you can chat with your cells if you want to. Pretty cool. Okay, so now let's talk about some modeling. So I described like the image processing, I described the cell tracking, but now we need to model our gene expression system. And I know Brian was here in the fall, so you probably saw some stochastic modeling from him. And this is largely going to be in the same vein, except for a little bit more put towards our system. I'm sure everyone here knows the central dogma of molecular biology. But we described these kinds of processes using what's called the chemical master equation. And it gives you this probability distribution over, say, number of protein or number of RNA, which is evolving over time. So we know there's heterogeneity in the system. We know that different cells are acting differently due to sort of fluctuations, diffusion, et cetera, et cetera. And we can model that using this chemical master equation. And the important thing to like, if you don't care about that, is that this gives you a probability distribution of, say, number of molecules, which evolves over time. And this is a useful thing to work with from a modeling perspective. The problem is that solving this system is computationally expensive. So here I'm just showing a big lattice of these dots, these nodes. And you can think of each node corresponding to a different equation in our system of equations. And as we add more and more species to our model, this grows bigger and bigger until my computer melts down and we can't do anything about it. So the way that we tackle this sort of computational challenge is through finite state projections. And this is basically a way to take that huge lattice that I described before and project it down to a finite set of equations. And the important thing to note is that you kind of make this sync state, which basically says, like, if I do a transcription reaction past 100 molecules, okay, the probability of having more than 100 molecules is just going to go into this state. And while it may seem like kind of arbitrary to do this, you can show mathematically that this sync state is it's like an error bound on your model. So you're keeping track of the error in your model in a way that lets you be sure that your model is kind of like true, compared to like just kind of doing something where you cut this off and pretend that the higher states don't exist. And we've done a lot of work. I've done a lot of work with Brian and Gregor looking at these errors and what you can do with them and what they mean, but I'm not going to go into that now. So let's do a model of this EL222 system. So we just modeled it at the level of abstraction that we chose was a two species system. So we modeled the EL222 abundance. And then we also modeled the fluorescent protein abundance, which I just denoted with protein here. And the way that we built this model is that you have a simple process governing the production and decay of EL222. This is a lie. We use something more complex than just a birth death process here, because we knew that the distribution of EL222 across the population has more variance than what you would get with this process. And there are ways to do that, but I don't want to go into. But it's not just this birth death process, something fancier. And then here we modeled the very simple model for the protein production, where you have some rate times the light. So this would be the sort of whether the light is there or not is U. And then EL222 is just the amount of this protein that you have. Now you'll notice that I did not explicitly include the dimerization of this molecule, which is because that happens super fast as soon as you shine the light. So we basically said that the amount of EL222 is in abundance, and it's roughly equivalent whether you're shining light or not. Okay, so this is another approximation which lets us do things that we wanted to do with our model. And then, of course, we don't observe just a number of protein. Like, we can't look at that fluorescent image and say, oh, we know that there's 10 proteins in there. We didn't do super resolution. We didn't have, and also there's like a bunch of molecules, so it doesn't really make sense to do that. So we used a measurement model that basically says for the fluorescent, the amount of fluorescence which you observe is essentially related to the number of molecules you have in the cell times some mean emission and variance in the emission of the fluorescence. And so this is a statistical model of that.
That's on a single cell?
That's right. Yep. So this is all we should be thinking all single cell stuff. Okay, so how well does this model do to describe the data? It turns out, this was obviously like not the only model we tried. We came up with the simplest one that was consistent with the data. And so this is whether or not this is an experiment where we shine the same light to every cell, and we wanted to calibrate our model. And so what we did is we have dark blue is lights are shining on the cells. Light blue is no light is shining on the cells. So we did an experiment like this, on, off, off. And the red traces are individual cell trajectories of fluorescence over time. And then the green heat map behind it is the probabilistic model that I showed you before, this finite state projection based model. And you can see that, so basically the darker the green, the higher probability we expected to see a cell with that amount of fluorescence. And you can see that a lot of the trajectories are falling in the dark green and few of the trajectories are falling in the light green. So qualitatively, this looks good. We did, obviously, in order to tune our model parameters, we did do like quantitative analysis of how good of a fit these are. And to sort of simplify and just give an eye check of how good the model is doing, we just look at, for example, the mean fluorescence from the trajectories, which is shown in the red, and the mean from our model. So we can take the mean from this green probability distribution, which again is evolving over time, and we see that it matches quite nicely. We can look at, for example, the variance. You'll notice that we have a big peak here, which we didn't necessarily capture, and you can kind of see that in the trajectory plots. You kind of have these. So it's not perfect, but it's pretty good, and it's at least capturing the sort of dynamics, like the timescales of the increase and the timescales of the decrease quite nicely. Now, of course, this is just a simple test on a relatively rich data set, and we were tuning parameters based on this data set. So then we did a validation where we just had light on all the time, and we see that again, the mean and variance match pretty well, but here we weren't tuning parameters. So a different experiment, different cells, but actually we can predict the behavior pretty well here, okay? Now, it's not super surprising, like this is a more complex data set that probably has all the same dynamics that this one does, but it's notable that this was between experiments and in fact, a different light profile, okay?
Is there any reason why data starts decaying at the end?
Yeah, so that's a good question. And I was looking, as I was going through these the other day, I was like, yeah, why does it go down at the end again? So we had a few things happening. The cells die at the end of the experiment because they make a monolayer and then they start to starve each other out. And so that's that decay is not really photobleaching. It's more of this effect. So really, in practice, we can only run these experiments about out to 600 minutes rather than sort of 750 or whatever.
So fluorescence emission of these proteins,
Mm-hmm.
is it near infrared or?
I don't remember what the wavelengths are. Yeah, I might have it in a supplementary slide, but yeah, there was. So I think what's maybe you're alluding to is when you're doing optogenetics, you have to sometimes worry about this blue, like the light you shine on the cells could stimulate the thing you're trying to, if the wavelengths are overlapping. And we didn't have any of that effect here. They were far enough apart. But it's yeah, I don't remember exactly do you know what it is, Gregor?
Like which protein is it?
It's that M scarlet.
M scarlet, Jason. Do you remember this? 650?
I don't remember off the top of my head,
Somewhere between 550 and 650.
Okay,
And the blue is which wavelengths?
I don't have that.
Okay, and so related to the blue,
Mhm.
ask back, okay. You get phototoxicity from the blue light. Can this also cause something like this? Because we know that when we do like Guppy stain, like we use Guppy staining, but use a fluorescent or violet light to basically excite that
Yeah.
we do this too often, or CFP excitation, you get phototoxicity.
That's a good question.
Because they are
We didn't,
absorbed
we didn't directly look at it. We did a lot of experiments where we looked at growth curves with or without blue light.
You
And
see the same thing if you don't shine light on it.
yeah, that's so we see we see similar population growth curves.
Yeah.
So they should be at least reproducing at the same rate no matter what. Yeah. Okay. So Now we're gonna go back, yeah, we're gonna get back to controlling these things. So we've shown that we can shine different light patterns, different light over time. Those different lights over time give different gene expression patterns. And now we want to make the cells do what we want them to do. So we define a set point, which is like desired number of molecules over time. We search over the space of input light sequences, where, you know, we just did a sort of what's like some people would call it like a bang bang controller, which basically means the light is either on or off. So it's a binary space, and you have some fixed number of time points over which the light can be on and off. And then we come up with, we used our model to basically say, okay, so yeah, this I want to emphasize something here. So when I say search over the possible input space, I mean we basically take all of these light sequences, we put them into our model, and our model will predict a curve which may or may not match the desired curve. And this was the set of light inputs that said, okay, if you do this to our cells, it's going to make a profile which matches the desired curve. And then we did the experiment using that light sequence. Okay. Now, this is in the situation where we're not shining light to each cell individually, we're shining the same profile to all the cells. And what you see is that the average fluorescence matches the target average pretty well. I mean, it's not perfect, but again, there's no feedback control. This is just use your model that I showed before, calibrated. How well can it track this target?
Is there any memory? Like if you have this dimer spines and then you take the light away, do they all fall completely off? And does the state of the promoter actually has enough time to switch back into the off state?
Yeah.
Or
yeah, so there were a lot of experiments that were done to look at the dynamics of that at the RNA single molecule RNA level from Kamasha's group, where they were seeing some, I think it was like 10 seconds or something that from the time you turned off the light to when the transcription would shut down. So it was pretty fast and pretty robust. Obviously, we're looking at, you know, M scarlet, so we have lots of delays in our system relative to that. So I just showed you that like this open loop population level control can do what we want, but we saw that there's kind of a problem in that like every cell, while the mean is matching, every cell is doing lots of crazy stuff. And so we wanted to think about doing the single cell version of this, where we use our digital micromirror to shine light on individual cells in a way that also accounts for the current state of the cell. So basically, you shine light, you see what the cells are doing. So you segment them, you calculate the fluorescence, you have a statistical model of how much protein is in there, and then you design your future feedback, your future light signal based on the current state of the cell. And so that's where this all kind of comes back around. I don't know. Okay. And the things that we did, which were kind of interesting, is we needed to integrate the data in a way that is sort of atypical or not just like basic control theory. We did something called a Bayesian filter, which allows you to use probability distributions, which are non-Gaussian. And then we built this model-based, so using that stochastic model to control each cell. We also did something called receding horizon model predictive control. So what this means is basically instead of trying to predict the entire light sequence that we should send to a cell at a time, we just go a little bit into the future. So we don't think about these super long time future, because we know that we're going to get a new measurement in 6 minutes and update what we're going to do anyway. However, we want to keep an eye on the future, which is like what I'm kind of trying to denote with this gray bar, longer than what we're going to use, because this allows us to kind of like, if you have a change, you can kind of start priming your system for that change earlier, or you're at least aware that that change in the target, the dashed line is coming. And then you kind of iterate this over and over and over. Okay, so this is coming through the Bayesian filtering. So this is our stochastic model. We basically have a way of propagating the uncertainty, our single cell uncertainty molecule distribution from time t-1 to time t, whether or not the light is on or the light is off. And it's worth noting that these computations are kind of non-trivial, and each cell has its own version of this, which means that we had to we had to write like a parallel processing so that each, because we wanted to control the protein production in like hundreds of cells, and each one of them has their own model. So it's like a very non-trivial computational problem. But anyway, so you can do that. You estimate the state from your most recent measurement. And that's where this Bayesian sort of update comes in. And then you need to plan for the future, and you need a cost function. And we have one. It doesn't rely on Gaussian assumptions, which is kind of the key point. And so what we can do is we can validate this in silico. So we do stochastic simulations of our model, which is the red line here. So it's like simulating one cell. And then we use our Bayesian filter and FSP-based model predictive control to design the light sequence as it's going. So this is exactly doing the model predictive, the receding horizon model predictive control that I described before. And then you can see that drives it to some level. And then here it anticipated that the target was going to go up, and it started shining light because it knows it's going to take the system a little while to respond. And then it starts to go up and it follows the target there.
That's a great basic question, but so if you're using the stochastic simulation for every single cell, where you're doing this, designing your feedback for your cell, is the reason that you wouldn't be able to use a simpler model like an ODE just for every single cell, I guess?
You can definitely use an ODE for every single cell. The idea is though that you want to capture, like, you might expect that the ODE for a single cell, similar to the ODE for the population, we saw that, yeah, the mean matches, but the variance is high, and you would expect the same behavior at the single cell level.
So this kind of approach, what is the limit in terms of time? I mean, here you have you have like 50 minutes roughly, okay, when you see the dashed line go up and you reach the maximum. So let's say you have a faster folding protein.
Yeah.
Like, do you have an estimate about what's the limit in terms of controllability?
That's a good question. So one thing that I'm looking at, we're doing a follow-up, is which strategy gives you kind of the optimal time to target, because this is kind of hedging its bet against the variance and like the time to target. Whereas if you just do like a full blast, you could potentially get to the target faster, but with more variability. And so there's a trade-off that's kind of intrinsic to the stochasticity of the system.
And you also see stochasticity in the protein folding?
I'm sure there is, but we didn't think about it. We have a deterministic folding time. And we can also use this kind of thing to estimate the hidden state. So like, we are measuring the fluorescent protein but we can also use this to kind of get at, we can look at what our model says was the probability distribution of number of hidden species of the EL222 in the system. So the red color map is the probability of a particular number of EL222 at each time point. And so yeah, we see that like the red trajectory is what was actually found. And you can see it's not perfect, right? So there's still a lot of variability. For example, the red trajectory does not follow the darkest color the whole way here. But again, this is a hidden species, right? We don't observe EL222 in our system.
Are protein concentrations normalized to size of the cell?
Oh, yeah.
How
So
do you take into account the?
yeah, we do normalize by the size of the cells here. And that's an important thing. We spend a lot of time trying to think like, okay, how do you do the normalization? Should you normalize? Yeah, by area, by whatever of the segmentation mask. And so in the end, what we do is probably rough, but it was good enough for what we're doing. It's a hard problem, yeah. Okay, so then we did it in real cells. So this is the, each row of this would be a different cell. And each column is like a, is the light signal at a given time. And this wave here is because new cells are being born throughout the experiment. So you can see as new cells are born, they are all like turn on the light so that they can get to the target. And what we see is that while the mean matches pretty well, we have less variance than we did in the population control case. And so we kind of went from something that's open loop model based feedback control to actual individually cells which are targeted under the microscope. And to kind of show what that looks like, this is a video of one of these control experiments where the cell, like the purple is just the cells, and then you'll see yellow flashes, and the yellow flashes are when cells are being shown light. So you can see each cell is getting a different signal, and they are targeted at them. And then at some point, we'll see them all flash soon. There. That was when the target went up. And then we're kind of trying to just maintain around there. And
So is the yellow actually representing the light?
that is the light, yeah. So we can actually measure, we took a picture when we did the photo simulation. And so you can see it actually highlights a lot of challenges that we went through, which was like, you can't just send light to the whole cell because it kind of bounces, and the light you send here will affect this cell, for example. So we have to do an erosion of the mask and then only send light to just a few of, and we don't have perfect, infinite resolution. We can kind of, I think we have about 20, pixels in some sense per cell. So yeah, so that is kind of like demonstrating what we did. And with that, I'll be done with this part. I don't know if folks have questions yet.
One question.
Mhm.
So right now you control the mean. For the reason you couldn't have had a standardization or any for like the first 10 minutes.
So I'm not controlling the mean. I'm trying to control the number of molecules in the cell, not the mean number of molecules in the cell. And so when we look at the loss function, too much machine learning to this cost function, you'll see that this is the target number of molecules in the cell. And it's not, if we want to do the mean, we could do that. Like it would just be a different expression here that we're trying to optimize for, or the variance like you suggest, or whatever. And I think those are interesting things to do depending on what you want to look at.
But imagine if the mean, instead of mean with a very small variation,
Mhm.
that'd be almost the same as setting the target number.
A mean with small variation would be like setting the target number. Yeah, I agree. Any other?
How many different proteins you are looking at? Like, it's a million?
Oh, like how many proteins are in the cells? Yeah, this is a good question. So we were estimating that there is on the order of like 5,000 or so, like on the high state, but it's just an estimate. We don't know for sure. Our model like tells us something about the number of proteins, but it's a bit of a model fit. It's not because it comes from this. There's this mu RFP parameter here, which describes the mean emission, and that was something we fit to data with our model. So that means that this could be, if you double this and half this, then you can, so n would be like the number of molecules we're estimating, and this would be the mean emission per molecule. So the mean fluorescence per molecule or whatever. So you could imagine that like just by making this thing bigger or smaller and doing the corresponding change here, you kind of can estimate different numbers of molecules in the cell.
like if you look at the fluorescence, like if the number of protein molecules are very high,
Mm-hmm.
then the intensity of fluorescence is very high.
Mm-hmm.
And if the number is low,
Mhm.
then it is very dim,
Mhm.
right? And do you have a reflection in your experiment, like computational experiments?
Mhm.
That's the case, or is different?
No, it's like that, yeah. We definitely, and we didn't see like saturation of the fluorescence for the ranges that we were looking at. I'm trying to think, there was something else that you brought up, which was a good point. So there are like really interesting ways that people try and estimate molecule number from these fluorescence things. I don't know if that's something you work on too, but I've seen like, I'm familiar with looking at the statistics in after divisions and trying to estimate like what Johan Paulson, I guess, does that kind of thing. Yeah, and it's a hard problem, right? So again, like this problem, it turns out, is more about dynamics of the process than it is about the number of what's going on. And the number, what we found worked pretty good, but it's probably not optimal.
So could you speculate a little bit about application of this?
Yeah.
And also how this could maybe be used to better understand biology?
The second part, I'll let you speculate on. The first, the first part, I think is, the sort of like one of the motivating factors of this work was to try and think about, people are designing genetic circuits all the time to like make a molecule or produce something of interest, like that synthetic biology makes stuff that's hard to make in cells, okay? And they build these gene regulatory circuits like the toggle switch or oscillators and all sorts of different stuff. But each of those things has to go through a super extensive design, build, test process. Half of them, more than half, 90% of them don't work. And the iteration time is super slow because you have to do all of the genetic manipulations every time. So what these optogenetics give you the opportunity to do is to basically modulate a component in those systems, which is kind of like doing, in some sense, a parameter sweep across your genetic toggle switch or whatever when you're trying to design a synthetic biological system. And so the idea was like this kind of technology, if we understand and can do stuff with it, you can design, those kinds of synthetic systems more quickly. And then if you wanted to like reintegrate them into like a functional like thing, you would know what parameters you need. Another thing that people are doing, which is not as much at the single cell level, is like optogenetic bioproduction. And something we did in this study, but I didn't talk about, was like controlling the number of cells which are growing in the population. So if you want to maintain a population of like cells which are, their job is to like proliferate, and another population of cells which is like to produce the molecule you care about, can you like tune the ratios of those two populations using optogenetics? And so that's the other kind of, so these are both in the more biotechnology sort of like synthetic biology domain, but I'm sure there are, like the discussion we had yesterday about using yeast, I think could be an interesting direction with this too. Okay. Any other questions? No? And then I'll be pretty quick through the next part. So this was work that I did at Los Alamos National Lab, and hoping to continue as time goes on here. So we know that like, so this is about protein-protein binding affinity. So we've known for a long time kind of like shape complementarity is a feature of proteins that bind together and where they bind together. And so like early days protein-protein interaction folks would try and characterize how characterize these shapes in a way that lets you like predict if two proteins are likely to interact and how strongly. So you kind of have this notion of like of structural complementarity, which is cool. And this goes back to like the early 90s, and they're like probably spent, 17 days computing this image on the right. So it's kind of amazing how far we've come in that sense. And then there's also like the sort of electrostatic interactions, which in thermodynamics that determine how tightly two molecules are bound together. And it's kind of thought of, well, at least like what people, when they're kind of coming at protein-protein interactions from more of like a data-driven side, it's like, well, these interactions should be determined by these types of complementarities loosely defined. And so what we wanted to do is come up with some machine learning, which could kind of incorporate those features about protein-protein binding affinities. And as you may be aware, like machine learning is taking over, and in a lot of structural biology, folks, are using these tools all the time. So these are just some recent, I don't even know how recent, what does recent mean? So this is like from the last like 4 years, I think, all of these, which are just papers that talk about like using machine learning, exploiting, there's AlphaFold, which kind of is more of the folding problem, but the rest of the papers are really about like predicting protein-protein interactions and trying to use some of these ideas about the geometry and electrostatics, kind of trying to inject them into their machine learning paradigms. And so what are the types of models that we should consider? They should be like rotationally and translationally invariant. So if you give me a protein structure of two things that bind, and then you give me another protein structure, which is the same one rotated, it should give me the same binding affinity. So no matter how you translate or rotate the data, you should still get the same answer. And rather than putting in a super flexible machine learning model that like has to kind of learn that, you can directly define a machine learning model which is invariant to rotations and stuff. Okay, so that's the first thing. We don't have a lot of data of protein-protein interactions. So from the PDB bind, there's like, so we need protein structures and we need experimentally measured binding affinities. Now this is probably where the project started to go amiss, because we assumed that experimentally measured binding affinities were consistent, which was stupid. Anyway, back to the modeling. So graph neural networks are a way to inject those invariances that I was talking about. To understand them in comparison to like something like a convolutional neural network, which maybe folks have seen before, typically for a convolutional neural network, you supply data which is regular, like an image. So you have pixels. Pixels are well defined, and they're every pixel has the same number of neighbor pixels. Whereas like we can view our protein surface as a point cloud, which is very irregular and kind of not the same at all. And so the data representation matters there. We can view this image where you have pixels, which are nodes, which have features given by like red, green, and blue. And every pixel is connected to its neighbor pixels, like I show in the top right corner there. And that's like that is exactly a 2D convolution, which is viewed through the point of graph neural networks. This is to highlight that graph neural networks are in some sense a superset of convolutional neural networks with a particular connectivity. But we're not restricted with the graph neural network frameworks to like this kind of regular data. We can build our graphs from our point clouds or from whatever. We can make node features like which reflect the physics that we care about. So in this case, we built node features which were related to the curvature, the local curvature of the protein surface, the Poisson-Boltzmann electrostatics, stuff like that. In social networks, you can make node features which are different stuff. And the sort of like beauty of the graph, or the theory of the graph machine learning and the graph convolutions is it kind of abstracts the convolution operator in the top to when you have different numbers of neighbors, and you have kind of like some metric of distance between you and your neighbors. It lets you do a lot of the same stuff, but in a way which preserves these invariances that I was talking about. So we took the protein structures from heterodimer pairs out of this PDB bind database. we generate a surface representation using Connolly, what's it called? Connolly surface, escaping me, Connolly something. The software is MSMS, maybe you've used that. And then, it's called a Connolly surface, yeah. Graphs for the binder and partner are then constructed from those surfaces. And we give a set of features to those nodes, and both types of geometric and chemical features may be relevant. I'm going to have to speed up. So so basically we built a neural network which does the following. You have a graph for the binder, a graph for the partner, which have to share weights. And you can think of this as learning the local fingerprints of the molecular surface. So you have there's no interaction between these two things. They're just kind of mixing those node features independently. And then we make a second graph, which is across the interface of the proteins, which is kind of modeling how the geometries and electrostatics are interacting together. And that makes a fingerprint for that. And then the last step is to aggregate all of the information into a number, like, for example, binding affinity. Now, again, with the sort of we should have known, we only had 967 after we did some filtering based on structural accuracy of the heterodimer pairs. We didn't want things that were obligate, just work together forever. And so we didn't have much data. And then if you look at the log KDs from that data set, we can see that it's a huge variance in the affinity values. I will say also, I am making fun of us for saying that we should have known there's only 1,000 data points, but one thing that injecting some of these physical features does, the invariances, is it does lower the data requirement. So we kind of tuned our modeling perspective based on this limited data availability. And then we wanted to see if before we start working on experimental data, we wanted to see if we can actually do basically fit these sort of delta G. So it's the delta G of the binder partner complex minus the delta G of each thing independently. And you can kind of get that from Rosetta scores. So I don't know if you all do you know what Rosetta is? Does anybody not know what Rosetta is? It's okay. I didn't know what it was before this project. Oh, okay. So Rosetta is a physics-based molecular modeling software that is used to design new proteins, among many other things, as well as analyze structural proteins. And it has been used as a guideline to design new stuff for a long time now. But it's not perfect. So basically, we found that our GNN can pretty well emulate what's going on with Rosetta. So whatever the computational pipeline, the complex biophysical modeling that does, we can do really fast with a graph neural network. So that was something maybe useful, but we already knew that Rosetta scores don't work good compared to experimental data. So the utility was limited. But then we thought, you know what, let's just go ahead and make the problem a little easier. So we split our data into 3 categories: high, medium, and low affinity binders. And then we trained our graph neural network to predict whether or not a complex was a high, medium, or low affinity binder. And we found that, so I'll just turn your attention to the plots, the accuracy plots on the right, and really even just the top right one. So from a random guess, high, medium, low would be 33%, right? We could get our model on test data up to around 45% accuracy, which is better than a random guess, but still pretty not good. So it wasn't, yeah, and this is kind of where we are. We need more data if we want to try and do better, or we need to rethink this component, because we know that this is 40 years of data from, some are using this isothermal telemetry, some are using surface plasmon resonance. There's structural uncertainty involved. It's super, super complicated. And it's kind of a miracle. I think, in my opinion, it's a miracle that it works at all. Okay. So this is obviously a ton of work. That wasn't just me. So Stephen, as I said before, did a lot of the work at Pasteur on the engineering side of the software and running those experiments. That was how we spent the first 6 months of COVID last. And then Yenting, who was my mentor at Los Alamos, and then lots and lots of other folks over the years who have contributed. And yeah, if you have any other questions, let me know. And now I have to recruit you. So we are hiring for applying at the postdoc research scientist level and also just like postmasters, whatever. To do kind of natural language processing on cancer data sets. We work with the National Cancer Institute. We have a bunch of pathology reports and we do cool science with them. So if that is something you're interested in, feel free to reach out. If you are going to apply, let me know. And yeah, I can make sure your application can be seen at least. And yeah, with that, I can take any other questions.